So chapter 4 is going to look at our genetic components of our cell. So you need to know what the structure of DNA is, and so DNA stands for deoxyribonucleic acid. That's the D, the N, and the A. So DNA is a double helix, and this means it's made up of two strands. And they form what's called a helix shape. So you can see one of the strands right here will go like that, and then the other strand is on this side, forms like that. And then this structure is our helix. If you were to untwist the helix, it would look kind of like a ladder where the rungs are in the center and then the two strands are on the side. So a nucleic acid, which is this monomer, is made up of three things. We have a phosphate group right here. So here's my phosphate group. We have deoxyribose. This is my sugar. And then I have a nitrogenous base. So the nitrogenous base is up here. All DNA has the same phosphate and the same sugar, deoxyribose. Deoxyribose is where it gets its name, deoxyribo nucleic acid. And the thing that's different about each DNA molecule is this nitrogenous base. This is the only thing that differs between our different DNA pieces. So there are 4 bases that are used in DNA. We have adenine, thymine, guanine, and cytosine. So adenine, guanine, thymine, cytosine. Uracil is not used in DNA. This is only used in RNA. So these base pairs, right, these A's, T's, G's, and C's, they pair up with each other in a very specific pairing mechanism. So A always pairs with T, and they have 2 hydrogen bonds that form this connection. And then G always pairs with C, and they have 3 hydrogen bonds. I kind of like to remember this too, because T and 2 sound, they both have that hard T sound at the beginning, and then G, C, 3, they all rhyme to remember the number of hydrogen bonds. So if you look at this, we have a purine, something with 2 rings, always bonding with a pyrimidine, something with 1 ring. And that helps to keep the DNA width, this uniform 2 nanometers apart, so that the ladder is straight, right? You wouldn't want a ladder that looks like this. That would be super funky. So by having something with 2 rings and something with 1 ring that always pair together, that keeps this width of the DNA always the same. And you don't have to remember the number, you just need to know that it stays the same. So DNA is made up of in discrete units called genes. So you have an entire genome with hundreds of thousands of DNA base pairs, but we only use a small percentage, less than 2% of that, for actual physical expression of our genes. So what we have is we have DNA, which is going to code for a kind of a temporary molecule called RNA. This RNA is going to be able to leave the nucleus of the cell and come out to the ribosomes and make proteins. And these proteins are what you see. So this is how your DNA codes for your physical appearance. But we have a huge amount of DNA inside of a single cell. And if you've ever packed anything to go like on a trip or anything, you know that you kind of have to pack things small in order to get a lot of stuff to fit into a very small space. So let's talk about how DNA is kind of organized. So DNA is organized into discrete chromosomes. You have 46 chromosomes. There's 23 pairs, so 23 from mom and 23 from dad. And these are highly condensed. A loose, uncondensed version of DNA is called chromatin. So let's start at the top. So at the top we have, it's the most uncondensed, it's literally just a single strand, or, a double-stranded helix. To make this a little tighter, what we do is we take these strands and we wrap it around these proteins. These proteins are called histone proteins, and this entire particle right here is a nucleosome. So think about like when you coil a hose. If you take the hose and wrap it around a coil or one of those kind of coiling machines, then it's going to create kind of a smaller package, right? We're going to put the hose away. We don't just leave it all out as a large strand. Those nucleosomes then coil around each other, and then they supercoil into this kind of wavy structure here. And then that wavy structure gets coiled up again. So as we progress kind of down this image, things are getting more and more and more dense. So they're getting packed closer and closer and closer and closer together until we get a chromosome, which is the most highly condensed version. So chromatin and chromosomes, they're both just DNA and protein. But chromatin is kind of loosely organized, and then chromosomes are very dense. Alright, so you have 23 pairs of chromosomes. You do need to memorize that. And when our cells are ready to divide, so when we have a cell that's maybe getting too big and it's about to split into two small cells, what we need to do is we copy the DNA. So we end up with these duplicated chromosomes like this. So this is a duplicated chromosome where the left half and the right half are exactly the same because they've been copied. And then eventually we'll split this down the center. One chromosome can go to a new cell, one chromosome can go to a new cell. And then we can have two cells with identical genomes. So when we look at this, we have two halves that are the same, and we call these sister chromatids. Each little finger is a chromatid. They are joined together at this kind of area where it necks in here. It's a little skinny there, and that's called the centromere. And on each side of the centromere, there's these little, they kind of look like kidney beans. There's these little proteins called kinetochores. So these structures are going to be really important when we look at mitosis and how these cells kind of split apart. Alright, so from DNA, we're going to copy into RNA. RNA. So RNA has a very similar structure of its nucleotides, but there are three very specific differences, and you need to know what these differences are. I always ask a question about this. The first difference is that RNA is only a single strand. DNA is a double strand. Alright, so here is my RNA, one strand. Here's my DNA, it's got two strands. RNA can be double-stranded very short amount of times. It can kind of fold back up on itself like this. But in general, RNA is one strand and DNA is two. We've already talked about these bases, uracil instead of thymine. So DNA has A, T, G, and C, where A bonds with T and G bonds with C. RNA has C, it has G, and it has A, but instead of T, there's no T here. It has U, uracil. So in RNA, G and C still pair together. That doesn't change. But in this case, A is going to bond with U with two hydrogen bonds instead of T. You can just basically take out the T and replace it with a U. So anytime you see a U, you're looking at something made out of RNA. And anytime you see a T, that's a dead giveaway that you're looking at something made out of DNA. And then they have a different sugar. DNA has deoxyribose, and then RNA just has ribose, which is why it's RNA instead of DNA. There are many different types of RNA. So rRNA stands for stands for ribosomal RNA, tRNA stands for transfer RNA, this is a tRNA right here, and then mRNA stands for messenger RNA. So one of the things that's really amazing about life is the genetic code, and that all living organisms use the same genetic code. So the genetic code is going to come in handy when we go in the second step from RNA to proteins. Right, so this is how we read, our body reads RNA into proteins. So RNA is coded into these 3 nucleotide sections called codons. So every 3 nucleotides is called a codon. And these codons then translate into the monomers of proteins, which are amino acids. You do not need to memorize all the amino acids, and you definitely don't need to memorize this chart. If you need to use it, I promise you I will give it to you. So the way you read this is you start with the first letter. You start with the first letter, and the first letter is going to be in this central circle. So this codon starts with a G, so I'm going to find G. Then you move to the second letter, and as you move letters, you're going to go out further. So the second letter is C, so I'm going to move out to C. And then the last letter is U. All 4 of these are here, so I'm going to move out to U. And then whatever amino acid is right next to that is what we're going to be coding for, and in this case, ALA codes for alanine. So pick one other of these codons and try it out on this codon wheel and see if you get the same amino acid as the image. So take a moment and reflect on the differences between DNA and RNA in your cells.